There is an organ in the human body that performs two completely different functions simultaneously. These functions are so distinct that, were it not for their coexistence within the same anatomical structure, it would be hard to believe they belong to the same tissue. One of these tasks takes place in the world of tubes, ducts, and digestive chemistry. The other occurs silently, in microscopic islands scattered throughout the tissue, regulating something as fundamental as blood sugar levels. This organ doesn't beat with the force of the heart, doesn't filter with the visibility of the kidneys, and doesn't occupy the popular imagination like the brain or the liver. However, without it, digestion ceases, metabolism collapses, and life, in its most literal sense, becomes impossible. It's called the pancreas, and most people don't think about it until it malfunctions. That in itself is one of the most fascinating aspects of this organ. Its functional invisibility produces no sensation when it's working correctly. It doesn't generate a perceptible pulse. It doesn't move the abdomen. It doesn't emit signals that the conscious body can interpret. It operates in absolute silence, coordinating two physiological systems of extraordinary complexity, and it does so every second of our lives. Understanding the pancreas isn't simply learning about a digestive organ. It's glimpsing one of the most elegant and, at the same time, most silently vulnerable systems in all of human biology. The pancreas belongs to that category of structures that classical anatomy was slow to fully comprehend. The earliest records of its formal description date back to ancient Greece, when the anatomist Herophilus, in the 3rd century BCE, identified a massive tissue in the abdominal cavity, without fully understanding its nature. Galen, the Roman physician whose authority dominated Western medicine for over 1,000 years, acknowledged its existence but attributed to it a secondary function, almost as structural filler. The idea that this organ played a central role in digestion didn't take scientific form until the 15th century, when Johann Georgsung described the main duct that bears his name, the duct of Wirsung, and it began to become clear that the pancreas was an active gland. However, the understanding of its endocrine function, regulating blood sugar, wouldn't arrive until much later. And when it did, it changed medicine forever. To understand why the pancreas matters, we must first situate it spatially. It lies in the abdominal cavity, nestled deep behind the stomach, resting against the spine, and in close anatomical relationship with the duodenum, liver, and spleen. It is an irregularly shaped, elongated organ that measures between 15 and 20 centimeters in length in an adult and weighs approximately 70 to 120 grams. It has a head, a body, and a tail. The head is nestled in the curve of the duodenum, the first section of the small intestine that directly receives the contents of the stomach. The body extends horizontally, and the tail points toward the spleen. This deep central position explains why the pancreas is so difficult to detect clinically. It cannot be easily palpated from the outside. It is surrounded by other organs and is only accessible through imaging techniques such as ultrasound, computed tomography, or magnetic resonance imaging. It's an organ that hides away, and this anatomical position has very real clinical consequences. Pancreatic diseases, and pancreatic cancer in particular, are often detected late precisely because they don't produce visible symptoms until advanced stages. Now, the external anatomy of the pancreas, while important, is only the beginning. What's truly revealing is its microstructure, because the pancreas is, in essence, two organs within one. And the way these two components coexist within the same tissue is one of the most ingenious solutions that evolution has produced in vertebrates. The larger component, representing approximately 85% of the organ's total mass, is the exocrine pancreas. The word exocrine comes from Greek and means that the secretion flows outward, in this case into the intestine through a system of ducts. Exocrine tissue is organized into units called acini, small clusters of cells arranged around a central space, like flowers around a point. Each acinus produces pancreatic juice, a mixture of water, mineral salts, and a set of digestive enzymes that are essential for breaking down food. These enzymes are themselves a biochemical marvel. Pancreatic amylase breaks down carbohydrates, converting complex starch molecules into smaller sugar units that the intestine can absorb. Pancreatic lipase acts on fats, breaking them down into fatty acids and glycerol. Proteases, a group that includes trypsin, chymotrypsin, and elastase, break down protein chains into individual amino acids. Without these enzymes, the digestion of the three major macronutrients would be severely compromised. Food would pass through the intestine largely unabsorbed. There is something particularly remarkable about how the pancreas protects its own tissues from these enzymes. Many of them, especially proteases, are capable of digesting proteins, and pancreatic cells are ultimately proteins. So why doesn't the pancreas digest itself? The answer lies in an evolutionarily sophisticated safety mechanism. Enzymes are produced and stored as zymogens, inactive precursors that are only activated once they reach the small intestine, where they come into contact with other enzymes that transform them into their active form. It's like manufacturing ammunition without a fuse, harmless until the precise moment it's needed. When this mechanism fails, the consequences are serious. Acute pancreatitis occurs when enzymes are prematurely activated within the pancreas itself, initiating autodigestion that can be devastating. The pain levels caused by this condition are among the most intense reported in clinical medicine, and in severe cases, the inflammation can spread to surrounding tissues with potentially fatal consequences. The pancreatic juice produced by the acini does not reach the intestine directly. It first passes through a system of branching ducts that gradually merge to form the main pancreatic duct, the duct of Unsung. This duct runs through the pancreas from tail to head, and, in most cases, joins the common bile duct, choledochus, the duct that carries bile from the liver and gallbladder, just before emptying into the duodenum through an opening called the ampulla of Vater. Bile and pancreatic juice reach the intestine together, complementing each other. Bile emulsifies fats, and the pancreas digests them. Many people also have a secondary pancreatic duct, called the Santorini duct, which opens independently into the duodenum slightly higher up. This anatomical variability is common and, under normal conditions, does not cause problems. But it can influence how certain disorders develop or are treated surgically. But now we come to the component that has marked the history of modern medicine in a way that no other discovery in digestive physiology has been able to match. Within the exocrine tissue of the pancreas, scattered like islands in an archipelago, are clusters of cells completely distinct from the surrounding tissue. They are called the Islets of Langerhans, named after the German physician Paul Langerhans, who first described them in 1869 while still a medical student. Langerhans observed these structures under a microscope. He noticed they were different from the surrounding pancreatic tissue, but he didn't understand their function. That took decades to be revealed. The islets of Langerhans constitute the endocrine pancreas, the second organ within the organ. The word endocrine means that the secretion goes directly into the bloodstream without passing through any ducts. And what these islets secrete has an impact on the entire organism that goes far beyond the local digestive process. Each islet is a small community of specialized cells. Beta cells, the most numerous, make up 60 to 70% of the total island mass. They are responsible for producing insulin, the hormone that allows glucose to enter the body's cells to be used for energy or stored as glycogen. Alpha cells, which constitute 15 to 20%, produce glucagon, a hormone with the opposite effect. When blood glucose levels fall, glucagon stimulates the liver to release stored glucose. Delta cells produce somatostatin, a molecule that acts as a local regulator, modulating the activity of alpha and beta cells. There are also other, less common cell types, such as PP cells and epsilon cells, whose functions are still being elucidated. What makes this system extraordinary is its real-time response. When a meal is consumed and blood glucose levels begin to rise, beta cells detect this increase and respond by releasing insulin into the bloodstream within minutes. Insulin acts like a key that unlocks the doors of muscle, liver, and fat cells, allowing glucose to enter. If levels drop too low, glucagon kicks in, raising them again. The result is a controlled oscillation, a dynamic equilibrium that keeps blood glucose within very precise ranges throughout the day. This feedback system is a classic example of physiological homeostasis, but its mechanical elegance should not obscure its potential fragility. When beta cells are destroyed by an autoimmune response, as occurs in type 1 diabetes, the body completely loses the ability to produce insulin. Glucose accumulates in the blood without being able to enter the cells, and tissues deprived of their primary energy source begin to metabolize fats uncontrollably. Without external insulin, this process leads to ketoacidosis, a medical emergency that, before the discovery of insulin in 1921 by Frederick Banting and Charles Best at the University of Toronto, was consistently fatal. The isolation and purification of insulin from animal pancreases, and shortly thereafter its synthesis and large-scale production, transformed type 1 diabetes from a death sentence into a manageable condition. It is one of the most important milestones in the history of 20th century medicine. In type 2 diabetes, the mechanism is different, but equally revealing. The body's cells lose sensitivity to insulin, a phenomenon called insulin resistance, and beta cells attempt to compensate by producing more of the hormone. For years, this overwork can maintain normal glucose levels, but eventually, beta cells become exhausted or decrease in number, and glycemic control deteriorates. Current research is intensively exploring the mechanisms that lead to beta cell exhaustion. Oxidative stress, chronic low-grade inflammation, lipotoxicity caused by the accumulation of free fatty acids, and glucotoxicity from prolonged exposure to elevated glucose levels. There is no definitive consensus on which of these factors is the most decisive, and it is likely that they all act together in varying ways depending on the individual. The blood circulation of the pancreas deserves special mention because it clearly reflects the organ's dual nature. The exocrine tissue receives its blood supply from the pancreatico-duodenal and splenic arteries. The blood it drains carries metabolic products to the portal system, which leads to the liver. The islets of Langerhans, for their part, have a disproportionately high capillary density compared to the surrounding tissue. This hypervascularization is not accidental. The release of hormones directly into the bloodstream requires immediate and abundant vascular access. The blood flowing through the islets captures the newly produced hormones and distributes them throughout the body in seconds. There is an internal architectural detail of the islets that research has been revealing with increasing precision in recent decades. Beta cells are not randomly distributed within the islet. In human islets, unlike those of some animal models such as mice, beta and alpha cells are more heterogeneously intermingled, with areas of higher beta concentration in the center and more peripheral alpha cells, but with a much greater interpenetration than initially thought. This cellular architecture appears to have functional implications. Beta cells communicate with each other through direct connections called gap junctions. This communication coordinates a synchronized secretory response, so the islet acts as a functional unit rather than simply the sum of individual cells. The innervation of the pancreas adds another layer of complexity. The autonomic nervous system, both the sympathetic and parasympathetic branches, sends fibers to the pancreas. Parasympathetic stimulation, mediated by the vagus nerve, promotes both exocrine and endocrine secretion. Sympathetic stimulation, on the other hand, tends to inhibit insulin secretion and stimulate glucagon secretion. This means that psychological stress, by activating the sympathetic nervous system, can have measurable effects on glucose regulation. This is not a marginal influence. Accumulating evidence suggests that chronic emotional states and prolonged stress can contribute to glycemic dysregulation, although the precise mechanisms and magnitude of this effect remain the subject of active research. The pancreas also responds to hormonal signals from the gut. When cells in the duodenum and jejunum come into contact with gastric contents after a meal, they release hormones such as cholecystokinin and secretin. Cholecystokinin stimulates the secretion of pancreatic enzymes. Secretin stimulates the production of the aqueous, alkaline component of pancreatic juice, which neutralizes the acidity of gastric contents so that enzymes can function under optimal pH conditions. Furthermore, enterocrine cells in the intestine release incretins, particularly glucagon-like peptide 1 and glucose-dependent insulinotropic peptide, which enhance insulin secretion in response to food. The existence of this enteropancreatic axis, this continuous hormonal dialogue between the intestine and the pancreas, represents one of the most active areas of contemporary metabolic research, especially in the context of therapies for diabetes and obesity. Pancreatic diseases, by contrast, reveal the silent importance of its normal function. Chronic pancreatitis, a frequent result of prolonged alcoholism or certain genetic predispositions, causes progressive destruction of pancreatic tissue, compromising both exocrine and endocrine function. Affected individuals develop exocrine pancreatic insufficiency, with difficulty digesting fats and significant weight loss, and frequently, over time, diabetes due to islet cell damage. Pancreatic cancer, predominantly ductal adenocarcinoma, is one of the neoplasms with the worst prognosis in oncology, with a 5-year survival rate that remains very low despite recent therapeutic advances. This high mortality rate is largely explained by the aforementioned diagnostic challenges and the tumor's tendency to invade neighboring vascular structures before producing symptoms. Current research focuses on serum biomarkers and high-resolution imaging techniques that allow for the detection of the disease at earlier stages. In the field of type 1 diabetes, the most promising scientific efforts are aimed at the replacement or functional regeneration of beta cells. Pancreatic islet transplantation, clinically established since the so-called Edmonton era in the early 2000s, has demonstrated that it is possible to restore insulin secretory function in selected patients using islets obtained from donors. However, the need for chronic immunosuppression and the shortage of donors limit its widespread application. Current lines of research are exploring the generation of functional beta cells from induced pluripotent stem cells, their encapsulation in devices that evade immune attack, and gene editing to produce cells resistant to rejection. There are preliminary results that the scientific community is closely monitoring. Although no therapy has yet progressed beyond the experimental phase for widespread clinical use, there is something about the anatomy of the pancreas that, viewed from a certain distance, reveals a deeper principle about the organization of the human body. This organ is, in a literal sense, an archipelago, a continent of exocrine tissue dotted with tiny endocrine islands. Each islet, invisible to the naked eye, imperceptible, without any sensation of its own to bring it into conscious awareness, performs a function that determines whether or not the body can utilize the energy from food. And each acinus, each secretory cell, each branching duct produces between 1 and 2 liters of pancreatic juice every day to make something as mundane as digesting lunch possible. The invisibility of the pancreas in conscious experience is, ultimately, a form of perfection. Systems that function well do not need to interrupt the flow of conscious experience to do their job. The heart beats and makes its presence known. The stomach growls, the muscles tire. But the pancreas simplifies its presence in the body to the point of being imperceptible. And it does so precisely because it is indispensable. It is one of those organs whose existence only becomes evident when it disappears or fails. And that absence leaves a devastating mark that cannot be easily compensated for by any other system. Perhaps that is the most revealing aspect of everything there is to understand about this organ. The human body houses mechanisms of extraordinary precision that operate tirelessly, relentlessly, and without complaint for decades. The pancreas is one of the best examples of this silent architecture. Two completely distinct systems, digestion and metabolic regulation, coexist within the same mass of tissue, coordinated by hormonal and nervous signals and adjusted in real time to every meal, every fast, every stress. All of this is happening right now, while the body continues its life without pausing to think about it. And that is probably the most accurate way to define what makes the human body still one of the most complex and surprising systems that biology has produced in 4 billion years of evolution. For thousands of years, the pancreas worked silently, without anyone knowing exactly what it was doing. Today we know that it supports 2 of the most vital processes in the human body, and yet it continues to operate without being noticed. That in itself says something profound about how the body is constructed. If this tour of one of the most underestimated organs in human anatomy taught you something new, like this video and subscribe to the Anatomy Revealed channel, where every week we explore what the body hides in plain sight.